Good morning, students. Today we are starting with our first chapter of class 12th biology, that is reproduction in organisms. First, we'll see the definition. Reproduction is a biological process of formation of new offsprings from the pre-existing organisms. And it is, of course, a vital process without which species cannot survive for long. And it ensures the continuity of the species generation after generation. Why? Because older individuals undergo senescence and they ultimately die, and reproduction helps for the survival of the species. Its another significance is it introduces the variation, which is of course the basic step of evolution. Before we start with the detail of different types of reproduction, we need to know certain terms. We'll start with the first term is lifespan. Lifespan is the period from birth to natural death of an organism. It varies from few days to thousands of years. Here in the table you can see that lifespan of different organisms, plants as well as animals, and this is very important that lifespan does not depend on the size of the organism. For an example, here parrot and crow, both are birds, right? Crow has a lifespan of 15 years and parrot has 140 years. See the difference. Same size bird, but the lifespan is very much different. We can also see the other examples of different organisms and their lifespan. One more example I can show you, that the banyan tree and the peepal tree. That is, banyan tree is Ficus bengalensis and the peepal tree is Ficus religiosa. Both have the same species, but look at the different lifespan of both of them. That banyan tree has lifespan of 200 to 300 years and the peepal, that is Ficus religiosa, has the lifespan of 2000 years. So there is no relation between the size of the organism and its lifespan. Now we will move to the phases of lifespan. Lifespan has 4 phases. The first phase is juvenile phase, of course, where the organism develops the capacity to reproduce. It attains maturity, sexual maturity. Of course, the second phase is the maturity, that is, it attains the reproductive capacity. The third is aging and senescence. Aging is actually the progressive deterioration of the body and irreversible changes which occur in body. That senescence now leads to the last phase of lifespan, that is death, where there is a permanent cessation of all vital activities. Now, in the next slide, we will move to the types of reproduction, that is the 2 main types of reproduction, which you have already also studied in 10th standard, the basic difference between the asexual and sexual reproduction. That is, asexual reproduction where only one parent is involved, no gametes are formed and no fusion of gametes occur at all. Parents and offsprings which are formed in asexual reproduction, they are identical, morphologically as well as genetically identical. Of course, asexual reproduction is the faster process, but one drawback, which is the major drawback, that no variation occurs in sexual reproduction. Asexual reproduction. In sexual reproduction, of course, 2 parents are involved. So the formation of gametes occur and the fusion of gamete, that is fertilization process, occurs. Then parent and offsprings, they are not identical because some events occur in sexual reproduction, that is the formation of gametes, where the crossing over and recombination process is the major step where the variation occurs. You have already studied in 11th standard, that is cell division process. This sexual reproduction is a slower process, but sexual reproduction creates variation, which is helpful in evolution, of course. And that is why sexual reproduction is better mode of reproduction and then asexual. In lower organisms, asexual reproduction is the common mode of reproduction, but on the onset of unfavorable condition, these lower organisms also switch over to sexual reproduction, so that when some variations occur in sexual reproduction, some variations will help them to adapt new features, and these new features will help them to survive in unfavorable condition. Now we will go to the detailed process of asexual reproduction and its type. Asexual reproduction, we have already studied that offsprings which are produced, they are genetically and morphologically similar, and that is why they are known as clones. One individual, that is one clone, is known as remit. Now we'll see the different methods of asexual reproduction. The first one is binary fission. Binary means two. Fission means breakdown. So in this method, a cell divides into two halves and grow into, these two halves can grow into adult organisms. Example, amoeba, like Paramecium, Euglena. One important thing we should discuss here is that binary fission is common in Protista and Monera, and we all know that Monera kingdom is of bacteria, cyanobacteria, and Protista, where the single-celled eukaryotic organisms are there. So there is some difference in binary fission which occur in Protista and Monera. In Protista and Monera, because both are single-celled, of course Monerans are prokaryote and Protista are eukaryote, but both are single-celled, and here these organisms when divide, this cell division is actually the mode of reproduction. And the important thing here is when the single-celled organisms divides, then the parent cell loses its identity. And that is why the single-celled organisms are known as immortal, because there is no natural death of the parent occur, because the parent cell divides and form two new individuals, and that is why it loses its identity. So no natural death is occurring here, and that is why they are known as immortal. Now we can see in this slide that the types of binary fission which are present. Binary fission, the cell can divide in different plane. The first one is, here is showing the binary fission in amoeba, where the irregular division occurs in irregular plane. The next slide is of Euglena, where the division is occurring in at the longitudinal plane, and the other example of longitudinal binary fission may be Trypanosoma, Leishmania. In the next slide, we can see that Paramecium. Here, the binary fission is occurring at the transverse plane. And another example of transverse binary fission you can see here is Planaria, which is of course platyhelminth. In Monera, that is the bacteria, when the binary fission occurs, there is some difference, which is some difference in the binary fission which is occurring in Protist and the Monerans. That in Monera, actually the binary fission is not occurring with the help of mitosis, but it is commonly known as amitosis. Why amitosis? Because there is no involvement of spindle formation in this mitosis. The single chromosome which is present in bacteria, here the diagram is of E. coli cell. The DNA gets replicated, then random division of the cell organelles which are present, and each chromosome will move and cell organelle will move to the daughter cell. Because there is no true nucleus in Monera, so there is no true mitosis occurring here, and that is why it is known as amitosis. In Protista, of course, mitosis occur, but the spindle is formed inside the nucleus only. As you can see in the previous slide, like in amoeba here, the spindle formation is occurring inside the nucleus. But in multi-celled organisms, the nuclear membrane disappears, as you all know, in prophase, and of course the spindle formation occurs after the disappearing of nuclear membrane. Now that's all for binary fission. Now the different type of fission here is multiple fission. In binary fission, we have seen that each karyokinesis is followed by cytokinesis. So the parent cell can divide and give rise to two daughter cells. But in multiple fission, it is important to understand here that karyokinesis is occurring. That is, nucleus is dividing so many times, but cytoplasm is not dividing. That is, karyokinesis is not followed by cytokinesis. So initially, many daughter nuclei are formed, which is surrounded by a common cell membrane, and then cytoplasm gather around each nucleus, the common membrane ruptures, and then each nuclei surrounded by cytoplasm will come out and form a new daughter cell, that is offspring. And this is also occurring in, of course, single-celled organisms. Here the example in amoeba. We have seen binary fission in amoeba, but the binary fission in amoeba is occurring in favorable condition, and multiple fission in amoeba is occurring in unfavorable condition. Here in this slide you can see that the amoeba, encysted amoeba is there. So 3-layered amoeba, 3-layered cell membrane is there. Inside that, the nucleus divide continuously and it forms the daughter nuclei, which are also known as pseudopodiospores. And when the favorable conditions arise, then the wall ruptures, each amoebulae come out and then form the young new amoeba. Multiple fission, another example, Plasmodium. Here the different stages of Plasmodium you can see in the slide, that is cryptomerozoid, right, which undergo multiple division in the shy-joint form. Multiple division occur in the nucleus and nuclei are formed, and then the merozoid stage where the daughter nuclei are coming out and forming so many Plasmodium. Of course, the Plasmodium you all know, that is the causal organism of malaria. So all these stages which are present here, it is occurring in human body. Like sporozoid enters in human body and then undergo multiple fission and form merozoids, cryptomerozoids, different stages. Another example of multiple fission here is another protist, that is monocystis. So you can see the monocystis and the various gametocytes are present inside the common membrane. Now the second type of asexual reproduction, that is budding. Here you can see a small outgrowth emerges out from the parent body and it forms a small bud. Now the nucleus divide, and out of the two nuclei which are formed, one nucleus, that is daughter nucleus, will move into that small bud. Now the bud is attaining maturity, and when it attains its full size, then either it get detached from the parent body or it will remain attached and it will form a new outgrowth. For an example, like in yeast, which is showing in the slide, yeast forms chain of buds. Now, can you tell me the difference between the binary fission and budding? See, in budding also, the parent cell is dividing and forming two daughter cells, but here the parent is keeping its identity. In binary fission, the parent loses its identity. And but here the small outgrowth emerges, and then it forms the another organism. So parent identity is retained here in budding. Now the third type of asexual reproduction, that is with the help of spores, of course, which are present in simple plants and fungi. And different types of spores which are formed, they may be endogenous or they may be exogenous in origin. Here in the slide you can see that Chlamydomonas, where the motile zoospores are formed inside the sporangium. You can see the other examples here like conidia, which are formed exogenously in penicillium. Then sporangiospores. Sporangiospores are also the spores which are formed inside the sporangium, but they are non-motile. Then another different types of spores, like here you can see the oidea, and another types of zoospores which are present here, sporangium, that is spores which are formed in conidio, which are formed on conidiophore in aspergillus. So the conidia are formed in aspergillus, just like penicillium. The fourth method, that is the gemmules. Now this is really very much important for you for the point of board exams also, because this is a very common question comes in board exam, that what is the difference in exogenous and endogenous budding. We have seen in yeast, and also it is present in hydra, that is the exogenous budding. Now we will study the endogenous budding. Small bud-like cells which are formed in sponges and which help to withstand the organisms in unfavorable environmental condition. These small bud-like structure which are formed inside the body of the organism, that is known as gemmules. So gemmules are endogenous buds. Now here during gemmule formation, the archaeocytes, because sponges are actually from porifera group, so they have the cells which are known as totipotent cells, which are stem cells and which can be differentiated into any kind of cells. So these archaeocytes which are present inside that sponges, they are loaded with food material. And the another type of cells which are present here, that is amoebocytes, they form a thick solid layer which is made up of chitin around the organism. During the autumn season, the freshwater sponge, the parent sponge, that dies and leaving behind a large number of gemmules. You can see in the slide in the picture, that is small round structures. So many round structures are there, and these are known as gemmules. These gemmules are produced by the sponges and they remain active by withstanding unfavorable conditions. So when the availability of abundance of water or arrival of favorable condition, then they start developing into new sponges. So it is an interesting method of asexual reproduction in porifers. Now we'll see the type of asexual reproduction which is present in plants. Especially asexual reproduction which is present in plants, it is vegetative propagation. Why it is known as vegetative propagation? Because new plant is produced with the help of vegetative parts of the plant. Now, what are the vegetative parts? That is root, stem and leaves. What are the reproductive parts? You all know that is the flower. But vegetative part, that is the root, stem and leaf, they can form the new plant. So the two types of vegetative propagation in plants, that is natural or artificial. Natural and artificial method. The first one is natural. We'll study the natural vegetative propagation, that is first one by stem. The stem may be underground or the stem may be subaerial, which some part may act as vegetative propagules. Now here I'm using the term propagules. Propagules is the structure which can form the structure which is present on plant which can form the new plant. So first we'll see the examples of natural vegetative propagation through stem. The first one is underground stem, that is the bulb of the onion. You can see here that fleshy leaves which are present. In the center of that fleshy leaf is the apical bud present, and the stem here is a disc shaped. Nodes and internodes which are actually get condensed and they form the a disc shaped structure. So in the center the apex is present, which can form the new plant. When germinate with the help of bud, then it can form the new plant. The second type, underground stem, that is tuber of potato. Now you can see here the tubers have so many eyes. In general term we use the eye. What is that eye actually? Eye actually is bud. Bud where the node region is there. Node region, on the node region the buds are present. With the help of this bud, potato, the part of that potato which is bearing that bud, it can form the new plant. The third example of vegetative propagation through stem is the rhizome. You all can, I know that you all have seen the ginger and then turmeric and all. When we see the structure of ginger here, the scaly leaves and nodes and internodes you can easily identify, because on the node where the buds are present, scale leaves are there. These scale leaves of course do not perform photosynthesis, and they are scale like. So at the corner of that scale leaf which are present the buds, these buds can give rise to new plant through vegetative propagation. The fourth example is the corm. Corm is actually the condensed rhizome only. The condensed rhizome where the nodes and internodes are condensed, and very common example of corm is colocasia, which is also known as herby, or jamaican, that is amorphophallus. Now these all were the examples of underground stem. Now the vegetative propagation through stem of subaerial. The first type is runner. We all have seen the grass. See, the grasses have nodes and internodes, and the leaf which are present on the nodes, they have at the nodal region new shoot which are present. When it touches the soil, then at that area only, if the bud is present, it can give rise to root as well as new leaf. So actually it is known as runner because it runs on the ground. Wherever the bud touches the soil, it can form the new plant. The second type is sucker. I think you all must have seen the mint plant. The mint plant vegetatively grow through the sucker. Here's the stem which has the node which is present below the soil, and from there the nodal region where the bud is present, a new shoot grows, and then it grows upward and come out of the soil. Again from the parent plant where the bud is present just below the soil, from there the new shoot arises, it grows obliquely and then come out of the soil. So this way mint grows. It is known as sucker. Stolon. You all have seen the the jasmine plant. Here what actually happens, at the base of the node which is present just below the soil, a bud which grows into a shoot, and then it comes out of the soil, and then it bends downward, again touches the soil. Where the node is present, the bud is present, it can give rise to new plant. You can see here in this structure that the base of the bud which is present, the base of the node where the bud is present, it can form the new shoot. That is the stolon. Now one important example, that is offset, where you can see the rosette of leaves and the tuft of root. Here is the example of an aquatic plant, that is Eichhornia. This iconia is also known as Terror of Bengal. You can see the next slide here. Here, the water hyacinth, Terror of Bengal. This Terror of Bengal water hyacinth, it grows very fast through offset. This offset is nothing but the branch which is coming from the bud, from the nodal region. So, and this water hyacinth, it was actually introduced in India because of its pretty flower, violet colored flower, but it created havoc here, and it is actually considered one of the most invasive weeds. It grows in stagnant water and it drains all the oxygen which is present in water body, and it leads to the death of aquatic organisms. So water hyacinth, where the vegetative propagation occurs at a phenomenal rate with the help of this offset. Now the vegetative propagation, next we will see the slide through leaves. You all know the very simple example, that is most common example, is bryophyllum, where at the notch of the leaf there is a bud which form the new sapling. Another example of vegetative propagation by leaves, that is begonia leaf and clencho leaf, where at the notch the buds are present, and these adventitious buds can give rise to root as well as new shoot and form the new plant. This new plant, a small plant, can be detached from the parent plant and it can form the new plant. Now vegetative propagation through root. We all know the sweet potato, Ipomia batata, where the adventitious buds present and they can form the new shoot. This is actually the tuberous root which store the food also, and it is used for vegetative propagation also. The next example is of Dahlia. Here also the adventitious buds which are present, which can form the new plant. Now here in this slide, besides this water hyacinth which grows vegetatively through offset, the another special type of vegetative propagation, it is through bulbil, and it is the example of vegetative propagation through bulbil. What is actually the bulbil? The bulbil is large fleshy buds. Here in this agave, the floral buds which are there, some of the buds can withstand in adverse condition, and when the favorable conditions reach, then these buds will germinate and form new plant. So bulbil is actually the large fleshy bud which can form the new plant, and which can also withstand in adverse condition, so they can act as mode of perennation. Now we have seen the natural vegetative propagation with the help of root, stem, leaf, then bulbil. Now we'll study the artificial vegetative propagation. The first method is by cutting. Cutting is actually the separated portion of, it may be from root, stem or leaf, and it is very common method which is used in sugarcane, roses, citrus, grapes, bougainvillea and other very common plants. The cutting is put into the soil, and then from that cutting which is actually present inside the soil, the roots will come out and the new shoot can grow. The second type of vegetative propagation, that is through layering. Layering is the method where the parent plant is allowed to develop roots while it is still on the still intact. Like on the parent plant, the new plant will be formed. It is of two types actually. If the stem is the stem can be bent and stem is weak. Then it can be bent and into the soil, as you can see in the slide, and where the stem touches the soil, a cut is made, and we can use the different types of phytohormones also, like auxin and cytokinin, and from the cut, from the incision, the roots and the new shoot will come out. But if the stem is hard and it cannot be bent, then we will use another method of layering that is known as air layering, and it is also known as gutti. Gutti, where on the stem the outer bark and the outer layers from the stem, it is cut, and you can see in this structure, air layering, the outer structure, the bark is actually the secondary phloem and outer tissue. It is removed, and along with the mud and manure, and it is sprayed with auxin and cytokinin. It is wrapped, this part is wrapped, the exposed part is wrapped with the help of all these material, and after some days we will see that the roots will come out. This type of layering is known as gutti or air layering. And the soil layering is of various types, as you can see, that is simple layering, then serpentine layering, then trench layering, then tip layering only, where the tip is, tip is there inside the soil, or the mound layering, where the part of the stem which is above the soil, a mound is formed, and this mound is formed with the help of manure and some fertilizers which can use, and it is sprayed, of course, with phytohormones. The new roots and shoot will appear. Now the next type of artificial vegetative propagation, that is through grafting. In this method, parts of two different plants are joined so that they will continue to grow as one plant. And of the two plants, the part which is present, which is actually in the contact of the soil, that part is known as stalk. We will use the stalk of the plant which has good root system, and the plant which is used for the shoot part, that is known as scion. The scion we will use from the plant which has got the good quality of fruits and seeds. Example where grafting is done is citrus, mango, apple. Now in this diagram you can see that different types of grafting is also there. That is the bud grafting, where the incision is made, and here the worth mentioning is that when the stalk and scion is made, the cut which is formed, that cut is generally formed at that is the cut which is formed, it is slant, so that the two parts, that is the stalk and scion, can be joined very tightly to join actually the vascular cambium which is present, because the supply of food as well as water, it occurs through vascular tissues. So the cambium and the vascular supply of the two different parts, that is stalk and scion, they are joined tightly so that there will be no interference with the vascular supply and the plant can grow easily. And this grafting is possible only in dicot plants because there is a ring shaped vascular bundles which are present. In monocot, grafting is not possible because the vascular bundles are scattered also and there is no vascular cambium present. You can see the different types of grafting which is there. It's a bud grafting where the small bud is used as a scion. A T-shape insertion is done at the stalk and this bud is inserted inside that T-shaped insertion which is made. Now here is the another type, that is tongue grafting. Now students, for today I think this much is enough. Revise all the slides And if you have any doubt, then you can ask me in the class group which we have made. In the next lecture, we will complete Reproduction Organisms Part 2, where we will study sexual reproduction, its various phases and its various steps. Thank you.